Hello, and welcome to the Select Science webinar titled Understanding Small Molecule Interactions Within Living Systems with Mass Spectrometry Imaging. My name is Anita Ramanathan, and I'll be moderating today's presentation. I'm delighted to introduce our speakers for today's webinar. Our first speaker, Professor Malcolm Clench from the Center for Mass Spectrometry Imaging at Sheffield Hallam University, will discuss the combination of mass spec imaging and three-dimensional cell culture systems. Our next speaker, Professor John McLean, Stevenson Professor of Chemistry at Vanderbilt University, will present technologies, methods, and results using mass spec imaging to study the molecular components of inter- and intra-colony chemical signaling and communication in bacteria. Just before I hand over to our speakers, I want to mention a couple of things. This webinar is as interactive as is possible, so please feel free to ask any questions at any time by clicking on the Ask a Question button at the bottom of your screen. We'll have a Q&A session at the end of this webinar. Now, without further ado, I'd like to thank both Malcolm and John in advance for their presentations. And with that, let's invite our first speaker, Malcolm.
Hello everyone, my name is Malcolm Clench. I'm Professor of Mass Spectrometry at Sheffield Hallam University in the United Kingdom, and I'm going to talk to you about some of the work we've been doing to use mass spectrometry imaging to look at 3D cell culture models, specifically living skin equivalents and tumour spheroids. The overall aim of the talk is to really present an overview of all this work and talk about why the combination of mass spectrometry imaging with 3D cellular models is a happy marriage, and then to talk to you about some of the progress we've made in not just using the technologies to produce pictures of distributions of compounds, but also to quantify both pharmacokinetic aspects of the interaction between xenobiotics and the models and pharmacodynamic responses. So the reasons for using 3D cell culture are in that they're a bridge between more conventional two-dimensional cell culture technologies and whole organisms for preclinical work. And we're talking here about looking at disease, in fact looking at toxicology studies, looking at pharmacology work. And we can see here on the slide they come between the 2D models and the organ models and the whole organism. And they certainly are useful for preclinical work, and there's definitely a role for 3D cell culture technologies in the 3Rs agenda, as in the reduction, refinement and replacement of animals in early stage work, and I'll talk to you about that a little bit at the end. So the first model that I'm going to talk to you about is a living skin equivalent model, and I'm going to introduce how the model's made and its relationship to skin before we actually look at the work we've done. So the skin is the largest organ of the human body. When you think about the skin, you'll be thinking about it as a barrier. You know, its role to sort of keep harmful substances out, and its role to keep all of your organs and things in. But it is actually an active organ. It is involved in the metabolism, the biotransformation of compounds. It, of course, is a route of excretion. Things are excreted through the skin. The sweat ducts in the skin are involved in controlling body temperature. It is an active organ as well as having a barrier function. It's of interest to the pharmaceutical industry as a site for drug delivery, and that's not just for drugs that are designed for topical application to actually treat disease of the skin, but also for transdermal drug delivery, for the delivery of compounds into systemic circulation, where you're wanting to avoid, for example, first pass effect. You don't want the compounds to pass through the liver. So this is human skin, and you can see, as well as cells, all the other components that go to make up skin. And I guess one of the things we're going to think about is the idea that these 3D cell culture models are models. The things that we'll be using will not be human skin. There'll be a model of human skin with some limitations, and there'll be tumour spheroids to represent solid tumours, not solid tumours, there'll be some limitations. And specifically, the limitations that they're going to be is there's not going to be blood vessels and the blood flow. There's not going to be nerves that can stimulate these responses, and there's no lymph function. So there are some limitations to the model. The actual living skin equivalent model is manufactured from two types of cells. So the 3D skin model is made from two types of cells. It's made from fibroblasts and keratinocytes. So the company we work with, Innovent, who make this model as a commercial product, they use fibrin as a scaffold. So initially, the fibrin scaffold is seeded with fibroblasts. They're left to establish themselves. And then once they're established, the second type of cells, the keratinocytes, are added. And then these two different types of cells, over time, self-assemble into the layered structure that is similar to human skin. So as I said previously, although it's not human skin, it does have quite a lot of the properties that are similar to human skin. So what's nice is, for a 3D model like this, is that we can create this air-liquid interface. So we do have a dry surface on the top. So we do have something that is sort of like a stratum corneum, so it is a barrier. We can use it to do drug penetration, and it can be colonized with bacteria. And it does express some of the key markers, protein markers, that you would expect to find in human skin. So in the next slide, we can simply see some H&E images of the the living skin equivalent model as it develops. And you can see up to day 12 air-liquid interface, where we've got this establishment of a dermis, an epidermis, and a sort of pseudostratum corneum on the top of the actual model. So the types of project that we're interested in are to do with interaction with xenobiotics. So here we can see a very simple study. So what we've done is we've simply taken the living skin equivalent model. It's been treated with the pharmaceutical we're interested in, and this is a project that's been sponsored by Croda, and they're interested in what happens when we treat skin with terbinafine, an antifungal agent, and if we add to the antifungal agent formulation one of their products, dimethyl isolyl orbide, which is a penetration enhancer. So in the initial mass spec image here, we can see a section, a vertical section through the skin that's been treated with terbinafine formulation and then sectioned after 24 hours. And you can see that this is a compound that's designed for topical use. It's designed to treat fungal infections of the skin. And as you might expect here, you can see that the actual compound is sitting in the epidermis with little or no penetration down into the dermis. So then what we want to do is compare the formulation with and without the presence of the penetration enhancer. And to do this, we need to do some quantification. So we wanted to actually put some numbers on it rather than just say more or less. So then we get into the issues around calibration and quantification for mass spec imaging. So we adapted a methodology that was reported by Vanderbilt in 2016, which involves applying arrays of calibration standards to your tissue using an acoustic microspotter. What you can see in box A here is the calibration array, which has been spotted onto the epidermis of the living skin equivalent. This is a control sample. And what we can do from that array of data, using the MSI-Quant software that was developed up in Sweden in Uppsala by Patrick Kalbach, is we can define regions of interest. It's very important when you're doing quantification in mass spec imaging that you matrix match the standards and the analytes. So here, the regions of interest have been defined as areas of equal size, specifically within the epidermis. And then those regions of interest were used to plot the calibration graph, which you can see in the inset box. Once we've got those calibration data, we can then extract regions of interest from samples that have been treated as controls or with and a small percentage of the penetration enhancer. And as you can see in box D, there is a clear increase in the amount of terbinafine in the epidermis if the penetration enhancer is used. And also, we can put some numbers on it using the calibration graphs shown in box B. The current work in this area is to validate these numbers by high-performance liquid chromatography mass spectrometry and see how the numbers we're getting from the mass spec imaging data stacks up to data from homogenates. But it's good here and to use the mass spec imaging because we're getting data specifically from the epidermis. As I said, the skin is actually an active organ and it is involved in metabolizing xenobiotics. In another project that we carried out in collaboration with a company called Phenatox, with Innovin again and with the University of Manchester, we were interested in how the model stacks up against human skin in terms of the presence of metabolizing enzymes, because there are lots of enzymes present in skin, as I mentioned earlier, and you can see those on the slide. So the analysis that we did initially was by conventional LC-MS-MS-based proteomics of human skin to look at what enzymes were present. So in terms of phase 1 enzymes, we could see no P450s in the skin that we were using, specifically in the epidermal region, so no phase 1 metabolites would be expected. But what we could see is a range of phase 2 type enzymes and things other than P450s. Specifically, we can see esterases and glutathione S transferases. And these were present in both the lab skin and in the human skin. But as you can see, these are at very, very low levels. So we were not expecting by mass spec imaging of proteins to be able to detect the presence of these particular enzyme compounds. So what we looked at was a way of a functional assay, a way of actually measuring their action. So we focused down initially on esterases because we had a probe for esterases. So methylparaben is used in LC-MS-MS as a probe for esterase activity. And you can see an esterase enzyme will simply remove this CH3 group here. So to carry out this experiment, we again treated the surface of lab skin with the methylparaben, incubated it for a period, sectioned it, and then examined the mass spec imaging data. And as you can see in the mass spec imaging data, where we're showing the methylparaben in red and the hydroxyl benzoic acid metabolite in green, we can locate within the lab skin section the presence of the green signal. We can locate the biotransformed compound, therefore we can confirm the presence of the esterase enzyme in the epidermal region of the 3D skin model. And again, this work is ongoing with a new PhD student starting to actually look for some of the other enzymes and can we extend this approach to detect the actual functionality of other enzymes in the skin. A project that we had sponsored as a UK knowledge transfer partnership between ourselves and Innovin was to look at how the 3D skin model works as a way of looking at wound healing. So these are the various phases of wound healing, and again you have to take on board that the 3D skin model is a model, so certain other aspects of wound healing wouldn't be expected to take place. We're not going to see any sort of immunological response or anything like that because those cells aren't present, but we would expect to see some inflammation. We would expect to see cells proliferating and migrating, and we would expect to see the actual 3D model remodeling itself and the wound healing. And we can see on the next slide how we went about producing the wounds. So we evaluated 3 methods for producing wounds in the 3D skin model. Those were a simple incision model, inserting a physical barrier, and burning the skin. And what we found was that in terms of producing a wound healing process within a reasonable time period that we could study, the best way to do it was as a simple incision model. And that is, we've produced a device that is able to actually stab the skin with a scalpel blade to a controlled size and controlled depth that we can then examine using the mass spectrometer or conventional staining techniques. If we examine the mass spectral data from the incision model, we can see that the dermal region, shown in mass spectrum A, shows quite a different spectrum from the epidermal region and the wound bed, which is shown in B and C. And as you can clearly see from these data, the spectra obtained from the wound bed are very similar to the spectra obtained from the epidermis, again confirming that it is cells from the epidermis which are growing down into the wound bed. If, however, we now look at an infected wound model, and this is the same 3D skin model that's been infected with S. aureus, we can see that even after 4 days, there is no wound healing taking place. We've still got quite a large open wound. And this very much mimics the situation with chronic wounds. You'll be familiar with chronic wounds and being one of the issues that's associated with an aging population, that they end up with large open wounds and ulcers that don't in fact heal. And this is because they get infected. But exactly the same thing is happening within our 3D skin model, which is giving us a nice platform to study the effect of infection on wounding. So now let's move on to our second type of 3D cell culture model. And these are tumour spheroids and spheroid aggregates, which is something we started to use in our own lab. And what we're trying to actually model here is a solid tumour. So in a similar way to the way that the living skin equivalent model isn't an exact match for a solid tumour, a spheroid or a spheroid aggregate isn't an exact match for a solid tumour. It's a model. But what it does have is similar features. What we're going to be missing are the blood and the blood capillaries. But what we do have is the actual oxygen gradient, which is a characteristic of solid tumours, from a well-oxygenated region on the outside, through to a hypoxic region, through to a necrotic region right in the middle of the tumour. So we've been working with osteosarcoma, which is a type of bone cancer. But it's a highly aggressive and highly metastatic tumour which has very low survival rates, and it is quite resistant to conventional chemotherapy. For us, it creates quite a nice model to study, not just because of the interest in the disease itself, but because while we're developing these techniques, the actual spheroids and the spheroid aggregates that you can grow from osteosarcoma are quite large. And what we've been doing is creating what we're calling spheroid aggregates or aggregoids. And the idea here is then that you grow up your normal tumour spheroids in alginate beads. And then after a period, we're going to get rid of the alginate. But the spheroids inside the alginate beads are your conventional small tumour spheroids. And you can see here in the staining the actual well-oxygenated and then the necrotic core, and get some idea of the scale. But these are, as you can see, are quite small still to be studied by mass spec imaging with any detail within the actual fine structure of the tumour. So what we've been doing is dissolving the alginate beads and then staining letting the actual spheroids aggregate together to form a larger structure. You'll see the actual small collection of spheroids in the alginate bead in the inset box here. But when we spin them down and let them grow together, if we then stain them up, you can see that the aggregoids, which are much larger now than the initial tumour spheroids, still have the same characteristic well-oxygenated areas, partially oxygenated in necrotic areas that we saw in the single tumour spheroids. And the difference is that they're bigger. And clearly also what's quite interesting here is that creating these aggregoids does promote and offer the opportunity for co-culturing, for growing up tumours that contain more than one kind of cell. So our initial work has been concerned with what happens with these aggregoids when we treat them with doxorubicin. So if you treat a 2D cell culture of osteosarcoma with doxorubicin, it responds to treatment. But you can see our 3D aggregoid does not. And so our osteosarcoma aggregates, our aggregoids, are a much better model of the real tumour situation in that they are resistant to treatment, which is what we were trying to achieve by going to 3D cell culture from 2D. So here's our first mass spec imaging experiment, and again it's a quantification experiment. So rather than use a calibration array created by spotting, here we've made use of the mimetic array idea that was initially produced by Steve Castellino at GSK. And that is, we've got, in our case, cell plugs rather than homogenates of the cells. We can use cell plugs. It's a single cell, it's a monoculture, so we can just grow up lots of these osteosarcoma cells, make them into a well array, spike them up with the drug, and then use that to produce a gradient of signal intensity to calibrate. We can see on the right hand box the doxorubicin in the spheroid, and we can see on the left just imaging ATP as a marker of what's going on in the cells. And because we've snap frozen these cells quickly, we've not killed them by adding the dox here. If, however, we actually work with a real spheroid and look at what happens as we increase the amount of doxorubicin in the spheroid, we can see, if we use the ATP as a marker of living cells or well oxygenated cells, we can see as we increase the amount of doxorubicin up to here in these cells that the ATP has disappeared, but the doxorubicin in green is increasing. And of course we've got a mass spectrometer. So we don't have the ability to image the drug. We again can use the mass spectral data and multivariate statistics just in the same way as we have done previously for human samples, if you ever follow our work, to actually look at what's going on within the spheroid. So here we're going to actually look again small molecule. We're going to use a discriminant analysis technique, PCA-DA, and we're going to actually look for things that are changing as we increase the amount of drug. You can see in these data that the different drug concentrations group differently. You'd expect that as discriminant analysis. We have told them they're different. But from the loadings plot, we can start to pull out signals that give us an indication of what's going on when we treat these spheroid aggregoid with 4 micromolar of doxorubicin. And then using a combination of accurate mass measurement and a bit of MSMS, we can come up with some putative identities for various compounds that we can see changing within the tumour spheroid. We can identify them, and then our current work is mapping these compounds that we've identified onto pathways and attempting to identify some of the other signals that we've definitely identified as changing with treatment. A real challenge in mass spec imaging, and particularly quantitative mass spec imaging, is to actually quantify protein markers. There's very, very little work in this area carried out using organic mass spectrometry. There's a bit with inorganic mass spectrometry and labelling antibodies, but little with organic mass spectrometry. So we were funded by NC3Rs, the Research Council in the UK that fund research that can lead to the reduction or refinement of animal use, to see if we could actually make the protein mass spec imaging genuinely quantitative. And I've put a health warning here on these next few slides, because this is definitely work in progress with no conclusion drawn at the moment as to how quantitative it actually is. But as we've seen from the previous sets of data, there has been success in using mass spec imaging for quantification. And I really do want to emphasize that, this idea that mass spec imaging isn't quantitative is an urban myth. For small molecules, there's a lot of papers out there showing how tissue spotting and the use of mimetic arrays can be used to generate good quantitative data from the mass spec images. So more of a question is, how can we do this for proteins? Well, we have been working very much with bottom-up mass spec imaging of proteins. That is, we carry out on-tissue digests and actually image the protein distribution by pulling in specific peptide signals from the M-tissue digest. So here we might pick this signal and we can actually produce a mass spec image of its distribution. And then there is some potential to identify the protein by carrying out an MSMS experiment on the peptide from the peptide mass fingerprint. But as those of you who have actually tried to do this will be aware, this is actually quite difficult at the moment, and the signals don't yield wonderful MSMS spectra. But this was the approach that we were going to use then, was to do an on-tissue digest, look at the distribution of the specific peptides in the tuber spheroids or the aggregoids, and use a combination of accurate mass and their ion mobility drift time to focus in on that particular peptide for quantification. And here is one of our spheroid aggregoids, and you can see we can, from this slightly squashed aggregoid, get some MS images showing the distribution of a range of peptides in it. They are quite abundant peptides at the moment from abundant proteins, but it's a way that we can actually make a start with this type of work on the spheroids. And we were very keen to actually, since the work was funded by NC3Rs, to actually try and use non-animal models to work up this particular technology. We started off thinking that the way that we were going to do this was by a variation of the Q-concat methodology that was developed in Liverpool. And in the Q-concat methodology, what happens is that the signature peptides, the proteotypic peptides that you wish to use, are all grown together to form together to create this Q-concat protein. So molecular biology techniques are used to create a protein that contains just the signature peptides that you're interested in one protein that can then be digested to use for standards. This was published 2013, so a few years ago now. And I would say, although this is really useful and we can still do it, the methodology for developing and making peptides has moved on. So you probably would actually more think about buying in a mix of peptides. So this slide shows what happens when you get to increasingly complex mixtures of peptides from our original Q-concat IMS tag protein with 12 peptides, up to a 200 peptide mix, up to 1,000 peptide commercially bought-in mix. And you can see the complexity of the signal that you get, and this issue we're trying to focus down in on a specific peptide where you need accurate mass at the very least, and we're hoping that the combination of accurate mass plus ion mobility will give us the specificity that we require. So the actual experiment we're going to do then is we're going to try and create our calibration array using cell plugs. I mentioned cell plugs earlier. And the reason we're using cell plugs is because there is a bit of evidence in the literature that if you homogenize the cells, it does lead to a bit of signal suppression. And that's certainly what we found for the peptides, that if we actually put peptides into a cell homogenate, we get a lower signal than if we put it into a mixture of into intact cells. So as I said, the way that we create our calibration array is to grow up lots of cells, to put them into wells created in gelatin as intact cells, to spike them up with the peptides, and then to section them as intact cells. You can see we get a much less signal suppression. Okay, so these are put into gelatin and then sectioned to give us our calibration array. And as we saw from the previous example with the small molecule work, we can use the MSI quant software to use these cell plugs to create the regions of interest, and then extract ion intensities from the regions of interest to create calibration data. So if we use MSI quant software, we can't use the mobility function that's available on the Waters Synapt instruments. So if we want to use the mobility function, then we have to use the Waters HDI 1.4 software, extract the intensities from this, and then go through Excel to create the calibration data. But that does give us the mobility function that gives us a bit of extra specificity. So what have we got? So here's just a couple of data sets. So if we run a calibration array and a spheroid on the same microscope slide, we get data that looks like this. We can put the imaging data. We can go in, pull out the intensities of various peptides, and look at how it's distributed around the calibration array and in the actual spheroid. That's what the data looks like. And if we use that on the simple calibration mix, you can see here that just by eye that we're getting something that looks like a concentration gradient. And if we actually extract the data, we can see that the calibration doesn't look too bad when you actually consider the complexity of the experiment that we've done to get to there. From the 200 peptide mix, we were actually able to generate decent calibration data for 26 prototypic peptides, so for 26 proteins in one experiment. And this is pretty much where we've got to at the moment in terms of the quantification of protein expression in the tumour spheroids. So to conclude, we think that combining mass spec imaging with 3D cell culture is a very happy marriage. We've got a whole family of different 3D cell culture models available. I've only scratched the surface here looking at the skin and the tumour models. And we can look at those with multiplexed imaging if we use mass spec imaging. I've talked a lot about animal reduction in terms of a reason for combining these two things, but a lot of the drive for the pharmaceutical industry at the moment is to actually reduce costs. And one of the big costs is late-stage drug failures. So if you can actually find out that there are problems earlier in the process by using, for example, a 3D model, and we saw how the 3D tumour model was a much better model than the 2D model, then there's clear economic advantages. I've demonstrated quantitative imaging of drugs and small molecules and shown that the mass spec imaging gives good data. And then we've started to look at can we actually extend this to do absolute quantification of protein responses. And these early data are quite promising, and that this work is ongoing. So what are we doing in the future? We've got a bit of work to do on the infected wound model, so we need to improve that, and then we can use the mass spec imaging to examine that. We need to tighten up some of these metabolites and lipids that we're seeing in all of these models and map them onto pathways. And for our protein quantification work, we are going to very much be looking at biopharmaceuticals, which obviously is a very hot topic at the moment. So it just remains for me to acknowledge the funders and co-workers who've taken part in these projects, because there's a whole lot of projects that I've discussed. So I wish to particularly acknowledge the work of Christina Russo, Emily Lewis, and Amanda Harvey on the skin projects. Yeva Kleberskaita, Laura Cole, and Rebecca Day, who unfortunately isn't in this photograph, on the tumour spheroid projects. Our external collaborators from various companies and institutions, NC3Rs and Innovate UK for funding, Innovan for the support that they've given us with extensive gifts of skin and advice over the years, and Waters for the opportunity to take part in this webinar. Thank you very much.
Hello, my name is John McLean. I'm Stevenson Professor of Chemistry and Director for the Center of Innovative Technology at Vanderbilt University. So for a number of years, we've been working in the area of untargeted molecular analysis, predominantly in building so-called human organs on chip. Now, the untargeted approaches that we use in those studies are equally applicable in natural product discovery. So for approximately 10 years, we've been working in developing strategies for discovering new natural products, primarily from bacterial systems. So what I'd like to tell you about today are some recent collaborations that we've had with Waters Corporation and being able to streamline and develop high-throughput strategies to make those discovery efforts even more efficient. Biological inspiration, we look historically at one of the earliest biological explorers, namely Charles Darwin. So Darwin said, it is not the strongest of the species that survives, nor the most intelligent, but the one most responsive to change. And so we use that idea in natural product discovery, where we explore extremophile organisms, and in our case, we explore cave-dwelling bacteria. So we use cave-dwelling bacteria as a source of natural products for several reasons. One is because if you're a bacterium and you live in a cave, you grow under constant light conditions, constant humidity, constant temperature. Now, if you also live in a cave, resources are very scarce. And so you have to evolve and develop strategies using basically chemical biological weapons against your neighbors so that you can keep all of the resources towards yourself. So for the past 10 years, we've collaborated with Brian Bachman, who's a molecular entomologist here at Vanderbilt University, and using these cave-dwelling bacteria that have never seen human organisms before as sources for these new natural product drug-like molecules. So to give you a sense of where we've been, and then I can tell you where we're going now, what we do is we utilize ion mobility mass spectrometry as a high-throughput strategy for broad-scale molecular analysis. And so one of the first questions we have to answer is, can we measure what we need? And so with the ion mobility mass spectrometry approach, we have the capacity to measure close to approximately 1 billion molecules per second. Now, this is a peak capacity calculation, so oftentimes in the data sets that I'll show you today, we're usually seeing somewhere on the order of about 20 to 50,000 molecules. Now, in an informatics sense, though, this puts a huge demands on the bioinformatics and biostatistical strategies because we can easily generate close to about a terabyte of data per hour per platform. Now, the second reason why we like the ion mobility approach is because early on we found that the different types of molecules, say lipids, peptides, carbohydrates, adopt structures or collision cross-sections which directly depend on the prevailing gas-phase folding propensity for those molecules. Restated a little more simply, the biological scaffold and the chemical scaffold that these classes of molecules use, they preferentially adopt very different structures. Now, this is tremendously advantageous for natural product discovery because oftentimes natural products that we seek typically have scaffolds that are very different from typical biological molecules. So if we can rapidly map where they occur in this collision cross-section mass-to-charge space, we can prioritize which molecules have unique scaffold structures combined with their biological activity in the drug discovery pipeline. So philosophically, this is a little bit like the famous physician Fritz Kahn. So what Fritz Kahn would do is he would take complex biological systems like those found in the human body and reduce those into a graphical and interpretable map. On the right, I'm showing a quote by Lewis Carroll, who's more well known as being a famous mathematician and also the author of Alice in Wonderland. And this sort of guides how we take this data organization approach. So Lewis Carroll said, the Cartographers Guild struck a map of the empire whose size was that of the empire and which coincided point for point with it. Basically, this is a map with a scale bar of 1 mile equals 1 mile. So clearly that's a useless map. So what we do is we use machine learning algorithms to let the data sort itself out. And so this is a little akin to the sliding puzzle game, which I'm illustrating here. And this is an approach which is used in a lot of internet commerce and other areas. For example, Amazon might try to make a prediction of what you would like to do in the future by making a picture of you, and that is represented by one of these tiles. What they'll do is they'll monitor your page views and your mouse overs and your buying history, and they'll create a tile. Then they'll say, we have 200 million customers, so we'll make a tile for each of our customers. And they'll start sliding those tiles around until they get a picture where they can find out which two tiles have the same types of behavior. Armed with that knowledge, what they'll do is they'll tell each of those two closest customers to do what the last person did last as a predictor of what they think that customer will want to do next. And so using a similar approach is how we organize this large-scale data that we collect. Now, for natural products, what that led to was a technique which we call bacterial fight club. So one of the problems in natural product discovery from things like bacteria is that you can look at the gene sequences and you can see silent gene clusters, but then a lot of empirical strategies are used to tickle the organism in different ways to cause the expression of those gene clusters. So one of the approaches that we did is we said, well, since we can use these machine learning algorithms, maybe what we can do is we can try to map out specifically what are the new molecules that are expressed when two monocultures are subsequently co-cultured. And so what I'm showing here, for example, is the ion mobility mass spectrometry data for an unknown bacteria, namely the CGS bacteria here. And we will create a monoculture of a bacteria we know a lot about, for example, Micrococcus luteus. Then when we co-culture those two species together, we make them compete for the resources. Under that competition environment, we hypothesize that the organisms will express some of those silent gene clusters, which could be bioactive compounds, which we would like to discover. And so if I show you the cultures, so they look sort of uninformative, as shown here on these plates. But now when we do the self-organizing maps on that complex data, what we're left with is on the right hand of that equation are heat maps of only those compounds which are expressed under the fight club condition. So what it allows us to do is go from approximately 20 to 50,000 molecules down to a few hundred molecules, which are those new drug-like molecules that we seek to discover. Now, an interesting approach with the fight club is now you can start switching out competitors. And so, for example, what I'm showing here on the left are using different challengers that are things like E. coli or subtilis and so forth, competing those against your unknown bacteria. And what you can get is you can get different expression profiles for different new natural products, which we can then discover. Now, all of these approaches required something like a static sample, and that is using tissue culture plates and standard cell culture conditions, scraping the culture, and then analyzing just those molecules, but losing all of the spatial context. So for approximately 10 years, we've been working on imaging mass spectrometry approaches combined with the ion mobility experiment. Now, this places some significant demands on the informatics as well. So what I'm showing here is on the top, in a typical mass spectrometry imaging experiment, you generate a matrix of mass spectra. On the bottom, what you generate is a matrix of matrices. But what it allows you to do is it allows you to combine a separation strategy, much like liquid chromatography, at each spatial pixel location. And so using these approaches, what we tried for several years to do is to use that to image under high vacuum conditions cell cultures. But we ran into a number of significant bottlenecks and challenges, one of which is when we would take the agar samples and place them under high vacuum, the samples tended to delaminate and fall off the sample plate, a little bit like a fruit roll-up. And so what we were really excited to do is to begin, through collaboration with Waters, is to combine DESI imaging mass spectrometry under ambient conditions to do natural product discovery. So to accelerate these types of analyses, we're combining the DESI with the imaging and ion mobility mass spectrometry. Using our imaging workflow, we're able to access the spatial location of each molecule and determine predator, prey, and predation within a single sample. Aside from spatial prioritization, our imaging workflow utilizes ambient sampling, such that our sample preparation is less than about 10 minutes without perturbation to the colonies within our system. Ambient sampling comes at a cost, though. While it's very rapid, it's complicated by requiring a firm substrate. So highlighted here are some of the sample preparation methods that we have explored, including drying the agar, imprinting, and growing on filters and membranes. For instance, Dorstein and coworkers have used imprinting methods in the first large-scale DESI co-culture experiments, which highlighted surfactant's presence in the predation of Bacillus subtilis. However, this methodology and others use a targeted approach, where we seek to use more of an untargeted approach. So what we found is that using a strategy first described by Baran and coworkers, is that using a nylon membrane is the most effective method for highly resolved and sensitive molecular images, in particular for these untargeted strategies. So what this involves is laying a nylon membrane on top of the agar and growing the bacteria directly on top of it. So the nylon membrane allows for unprecedented sampling of these different microbes. So not only are the microbes unperturbed during the sampling, but they're also prepared in less than about 10 minutes. So this method offers a holistic analysis of the environment of the microbes. Cells are grown on the membranes on top of the agar and the growth media. And the membrane serves to allow nutrients to pass through to the microbes, but it retains the colonies on its surface. We have also started annotating and tracking natural products involved in signaling within the environment of the microbes by sampling the areas outside of the cell colonies, but still directly on the membrane. So this is one of our first images with the desired spatial prioritization representing prey, predator, and predation. The three features are represented by the unique colors of these different regions of interest. It's important to highlight that our analyses do not encompass the three regions and three features, but rather these are just representative of many regions made up of thousands of features. To validate our method, we look to other more traditional natural product discovery workflows. So in one case, Rolf Mueller and his group, who focus on natural products from myxobacteria, reported close to 67 unique core structures with some 500 different natural product derivatives. So using a database that we created of about 300 of these natural products, we were able to identify the natural products within the same species, as well as a new strain of bacteria isolated in a quick cave environment. Annotating similar natural products for each representative species validated that our sampling was similar to that of other workflows. In the bottom right, we highlight one of our more powerful annotations, and this was namely DK xanthines. To give another illustration of this type of approach, we can use these heat maps to represent our basic schematic of the predator, prey, and predation regions. But we also soon realized that the Fight Club or predation is more complicated and dynamic than what our basic model suggested. These images highlight the dynamic nature of each of these features within each image. So as you can observe, this unknown M over Z in the bottom right travels across the image towards the prey in a dynamic way. The intricacy in these features, as well as other thousands that we were able to annotate, allow us to use statistical analysis and prioritize these new molecules in the drug discovery workflow. Principal component analysis can also be used to reduce the dimensions and determine commonalities between the samples in large sets of data, which is what we did for this particular image. The unsupervised grouping of the regions represented by their colors within the image show the chemical differences between each region. However, this grouping revealed information that was given to us within the spatial dimensions of the image itself. Now, we then turn to mass spectrometry imaging in order to take advantage of all of the other types of information contained within these images. So using imaging analysis, we were able to achieve an unsupervised grouping of the regions we previously highlighted in our heat maps. This annotation lets the intensities and spatial location of each feature dictate the important regions or segments within each image. We can then use these regions in order to spatially prioritize natural products or features of interest within each experiment. With this imaging analysis, we felt confident in the method that we had developed, that it was conducive for our untargeted discovery efforts. Most importantly, the workflow that we have developed is accomplished in approximately 3 hours per sample, and I've highlighted several of the times for each step in the analysis in the top part of the slide. Now, we do believe there are some streamlining places where we can get over those bottlenecks. But right now, 3 hours is much faster than how we could do our previous analyses using a static methodology. So this rapid sampling has momentous application in high throughput screening. To prove our method capabilities and screening workflows, we compare and annotate features of interest between two strains of myxobacteria, a cave strain and a laboratory strain. Within the top 100 features, within the respective predation zone for each of these bacteria, we find about an 11% conservation between the two different strains represented in the Venn diagram. This number is significantly higher than 3% conserved within all of the features of each experiment, not just in the predation region. Here we highlight unknown features that are both conserved and unique within each experiment. Each has heat maps that are of particular interest within the predation region itself. We aim to annotate and identify these features using the union of all of the dimensions within a DESI ion mobility mass spectrometry experiment. We are able to prioritize the features on the basis of their spatial location. Using high-resolution mass spectrometry, we discern molecular weights and formulas. In our initial ion mobility experiments, we were hopeful of different scaffolds resulting in different mobility mass correlations. So we were excited to see that in some cases this is true. Here, we represent a family of unidentified natural products which also occupy the same spatial location, but show one of these mass mobility correlations. Using tandem mass spectrometry, we're able to confirm that these molecules all had the same conserved structural moieties, which we're now using to help identify these features of interest. It's been exciting to share our work with you on this approach. Now, through this presentation, we wanted to highlight the ability to rapidly sample microbes within a holistic environment. The imaging capabilities allow us to use spatial prioritization to annotate these natural products of interest. Using these capabilities of DESI, ion mobility, mass spec, we can begin to identify these features of interest in a high throughput strategy for accelerated natural product discovery. So I would like to thank you for your attention, and I'd like to conclude by highlighting those individuals that contributed very significantly to this work. So almost all of the analytics that I've shown here were done by Berkeley Ellis in collaboration with Brian Bachman and Caleb Fisher. And I would also like to pay special acknowledgement to our Waters collaborators as well. So Dr. Mike Beatty and Dr. Roy Martin. And thank you for your attention.
Thank you, Malcolm and John, for your informative presentation. Now let's move on to the final part of today's webinar, the question and answer session. We have lots of great questions, so let's get started. Our first question is for John. The question is, How difficult is it to get different types of bacteria to grow on these membranes? And have you tried many other types?
So that's an excellent question. And what we've done is we've been using this workflow for a couple months now. We've looked at around 15 different strains. Some are gram-positive, gram-negative, eukaryotic. And we've only encountered one challenge. Everything seems to grow well, except when the cells are when the bacteria are stressed and they clump and they don't form a very flat biofilm, which sort of negatively impacts the imaging itself. So as long as we can grow them under conditions that they grow a nice film, we've had great success thus far.
All right, moving on. Our next question is, how do permeation rates through lab skin compare to real skin, specifically water, hydrophilic, and lipophilic molecules?
Yeah, that's a good question. And it's back to the idea of this being a model rather than human skin. So we need to understand it does behave slightly differently to human skin, and in this case it's particularly for hydrophobic molecules where the rate of penetration is much higher than normal human skin. But you just need to understand these properties of the model to make it useful.
All right. Our next question is for Malcolm. The question is, when you compared wound growth tissue to dermis, epidermis, do you know what was causing the signals that allowed you to determine their identity?
Yeah, there was a lot of signals that are different in the wound bed. We can see metabolomics analysis using the accurate mass of the signals, and then obviously MSMS to tighten up their identification, and then feed those data into pathways software to actually come up with an idea of the sort of pathways that are actually being stimulated. And in that particular experiment, we could see fatty acid biosynthesis and ubiquinone synthesis being upregulated. But if you want to read a bit more detail about it, the paper is actually just on the early view for the International Journal of Cosmetic Science, and all of the IDs of the upregulated metabolites are listed in that, along with the pathways.
All right. Thank you. Our next question is, do you plan to combine lattice light sheet imaging with mass spec imaging in the future?
John, is that one for you? We
so,
don't hear.
Yeah, so I think that's a great question. And sort of in a previous lifetime, I used to work a lot in more things like imaging velocimetry and building light sheets and things like this. I think there are a number of correlation issues in combining those strategies, but I think depending on the question you're asking or the instrumental approach, you might be able to do that in some sort of good applications. I don't know if, Malcolm, you would like to contribute to that.
No, we've certainly not used lattice light imaging at all. We do try and use multiple imaging techniques and find that having multiple sets of data helps us to understand and get more information out of it, but not that specific technique.
All right. Our next question is, does ion mobility help quantitative measurements on peptides? And does using ion mobility spectrometry help or hurt dynamic range and sensitivity?
Okay, I'll answer for the help. So what we've found is when we've been doing the quantification experiments, that adding in the ion mobility dimension to the complex data sets has helped the linearity of our calibration responses. And we think that's just, is due to the fact that we are improving the specificity over simply using mass alone. No effect on the dynamic range that I could speak for, and sensitivity we haven't really compared.
Lovely. Our next question is for John. It's a two-part question. The first part is, have you used nylon membranes in DESI imaging? And the second part question is, have you ever used fungi for DESI imaging? Can we use fungi in DESI IMS, although they produce spores and may contaminate the system?
So that's an excellent question. So we do use nylon membranes quite a bit, and we have done fungi. we have not noticed to this point in our systems in doing the fungi that we've had sporulation. So that could be something that people run into, but we have not sort of had to overcome that at this point.
All right. Our next question is also for John. It says, outside of GNPS and methylene, how do you determine the true structure of the induced secondary metabolites without scale-up and purification for NMR of every single compound?
So I would love to be able to answer that in a sufficient way, because then I would be an extraordinarily rich individual. And I'm rich in friends, I think. But the answer is it's very difficult. We have done a lot of work in big data and informatics, and what we can do is we can take any particular natural product compound sort of down a path with the MS/MS and with the high mass accuracy and the mobility. We can usually get a new natural product down to about 3 to maybe 5 different compounds, which it could potentially be. But we too are limited, just like many others in the field, where for us to get things like stereochemistry and to really restrict us from that 3 to 5 compounds to what the actual structure is by using NMR.
All right. Our next question is, in case the sample is crude in nature, like a multiple unknown organisms all in one contaminated site, is it possible to study them using DESI-MS?
I'm sorry. So if I interpret that question, can we sort of can we tease apart which of the different actors are contributing to the signal? Is that how you interpret that question?
I guess the question is about if the sample itself has multiple unknown organisms in it. So the sample is kind of crude in nature. So the question is it possible to study a crude sample using DESI-MS?
So the answer is absolutely. And I think the real problem comes in. So we've been also pushing some of these strategies into microbiome efforts. The real question becomes, how do you tease apart who has said what chemically? And so, in some cases we complement these studies with things like 16S sequencing. But what we also would like to suggest is that this Fight Club idea is really creating a biome in the true sense of both definitions of that word. It's 2 ohms, and it's also growing sort of in a community. What we've also done is started to do more complex samples, not totally crude samples, but things like 3 actors and scaling up in that direction, where we have a sense of how many different types of bacteria are all communicating at the same time, so that we can tease apart, ultimately, we hope, the different cues and communications that are given off by a much more crude sample.
All right. Next question for John. Did you try IMS with actinobacterial or fungal cultures? If yes, is the resolution good?
Yes, we have to both, and we don't see any limitations in the resolving power.
All right. That's all we have time for today. Thank you, Malcolm and John, for your discussion and the presentation, and thank you, all of our attendees, for your lovely questions. I hope you found this session valuable. I see all your questions here, so I promise to follow up with the speakers personally. If you have any more questions, please feel free to email me at anita.rama@selectscience.net. In just a few days, the on-demand link for this webinar will become available, so you can listen in again or invite a friend or colleague to listen. Once again, thank you for joining us, and I'll see you next time.